Hello, my name is Christy Amobi, and I work with Extra. Welcome to our education event. Today, I'm delighted to introduce Dr. David Fernandez-Antoran of the Gurdon Institute at the University of Cambridge in the IIS-Aragon in Zaragoza, Spain, who will discuss ionizing radiation in cell competition.
Hello, good afternoon, Christy.
As many of you know, ionizing radiation is one of the most common environmental mutagenic agents that promotes tumor formation by damaging DNA, but also forms the basis of therapy for the majority of cancers. Today, Dr. Fernandez-Antoran will discuss his group's research to unravel the molecular responses and cellular interactions that control normal and mutant cell behavior using the Extra 6.1 and 6.2 cabinet irradiators. I have a few housekeeping items to cover before I hand it over to Dr. Fernandez-Antoran. First, this session is being recorded. It will be available on our website and will also be distributed to you via email after the conclusion of the event, if you'd like to review it at a later time. Also, today, we will have a Q&A after the presentation. This Q&A will be a moderated Q&A, and I encourage you to share your questions at any point using the questions function of the GoToWebinar control panel. The session is approximately 40 minutes with questions at the end. Thank you so much for your attention, and with that, I'll hand it over to Dr. Fernandez-Antoran.
Hello, everyone. It's a pleasure for me to be here today. I'm going to try to talk to you about today the effect of ionizing radiation in cell competition, which is the main topic we study in my lab. So as many of you know already, we humans have our tissues fully populated by mutant cells. So we don't have wild-type cells as we thought at the beginning in our tissues. Our tissues are conquered by mutant cells, and these mutant cells basically are fighting for the space in a process called cell competition. Some of these mutant cells, or some of the cells, harbor mutations that are oncogenic mutations. However, they behave as a normal cells. So this is just an example, just to show you how a typical human epithelium of the esophagus looks in terms of mutations. You can see here, for example, the dots, the colored dots, depending on the age. You can see how these dots represent the clonal composition of mutant cells in our tissue, in the esophagus, in 1 square centimeter. As you can see, the older we get, the more mutations we accumulate, and the bigger these clones of mutant cells are. My lab is heavily interested in understanding how these mutations are related to cancer development. We are trying to understand how this fight between different mutations, different mutant cells in our tissues is shaped or is affected by environmental factors. So basically, this fight between these two populations of cells follow a typical Darwinian evolution laws. So as Darwin and Wallace was mentioning, the best adapted will survive and reproduce. So for the cells, as you can imagine, well, first of all, in nature, you can see when you have two different species and they are basically competing for the resources, those species that are more adapted to the environment will survive and reproduce. In the case of the tissues, in the case of the cells, it's exactly the same. So we have different population of cells in our tissues. Some of them are wild-type cells, but many of them are mutant cells, and they are harboring different type of mutations. And when we expose these tissues to specific selective pressures, which basically can be an environmental factor like smoke, drinking, even different chemical therapies like radiotherapy or chemotherapy, we are changing the rules under which these cells are fighting each other. And basically, those cells that are adapted to the new environmental conditions might be selected and basically overspan and basically overtake the whole tissue. If these selective pressures are maintained, we can end up with a huge increase in a specific population that can be mutant because it's the fittest population in this sense. And this mutation can basically accumulate other type. These cells can accumulate other type of mutation that basically can give rise to tumors. In my lab, we're trying to understand one specific environmental factor that humans are exposed to, which is ionizing radiation in all the ways we are exposed. basically mostly like clinical treatments, like radiotherapy treatments, X-ray plates, or CT scans. I'm going to show you first a project which I did during my postdoc at the Sanger Institute, when we demonstrate that ionizing radiation, even low doses of ionizing radiation, have an effect on the expansion of these mutant populations in our tissues. So basically, focusing on ionizing radiation, just let me show you basically what is known about radiation biology. So the radiation biology questions about ionizing radiation always have thought that the linear non-threshold model, in which the higher the dose of radiation we are exposed, the higher the damage, is the one which is used basically worldwide to use in protection regulations laws. But there are other type of models in which people, for example, the threshold model, think about there is a small threshold of radiation or dose which is not harmful to tissues. So we can be exposed to these doses, this small dose of radiation, with no effect on our tissues. There is also a bit controversial model which is called the hormetic model, in which even low doses of radiation can be beneficial for the tissue. Also, there are other models which might involve the type of data we have generated, which basically means that low doses of radiation might be even more dangerous than high doses of radiation. So basically, we use mouse esophageal epithelium as a model to study how ionizing radiation affects cell competition in tissues. And why esophagus? Because the mouse esophageal epithelium is a very simple tissue. It consists of layers of keratinocytes, and it lacks any other structures like the skin, for example, with glands or hair. It basically consists of layers of keratinocytes, and the proliferation is confined to the basal layer. Those basal cells, sometimes they exit cell cycle and they migrate to the surface in a process called terminal differentiation. So basically, this is a typical 3D confocal view of a mouse esophagus epithelium. And you can see how the basal cells at the bottom and the layers of differentiated cells on top. This is a very simple model, but very powerful to study basically cell competition mechanisms in vivo. So we use a very well-known mouse model for tracing cell behavior, which basically allows us to induce the expression of a reporter of fluorescent protein in our basal cells of the esophagus and follow them. So we can understand what these cells are doing depending on the condition. This mouse model was published in my previous in my lab at the MRC Cancer Unit in Cambridge in 2012 by Dupré and Alcolea in Science. And basically, they demonstrate using this technique that the mouse esophagus is populated by a single progenitor population, which basically tilted their behavior between proliferation and differentiation in order to maintain tissue homeostasis. So basically, this is a typical view of a clone. When we induce these clones in the tissue, we can follow the progeny of these clones. We can see all the cells that give rise to the or have been given rise from the beginning, from the initial label cell. So we can see clones that basically are maintained. We have basal cells and suprabasal cells, but we can see also some clones that are basically what we call floating clones, because all the cells in the basal layer have been committed to differentiation. So basically, this clone will be lost. There is a balance between these two types of clones in the tissue, basically because they need to maintain the tissue homeostasis. This is very simple to explain, because if we have cells that are proliferating in the basal layer all the time, and we have no correlation with differentiation, we can increase the number of basal cells, which basically might develop, might become a tumor. But in the other way, if we have too much differentiation, for example, we can lose the tissue barrier function and tissue integrity, which basically promotes a destruction of the tissue. So there's a very fine tuning mechanism controlling this behavior of the stem cells in this type of tissue. Basically, give us this average division outcome in which we have similar numbers on average of proliferating cells and differentiating cells in the mouse esophagus. So keeping that in mind, we use this model to study how ionizing radiation might be affecting the behavior of these stem cells in our tissues, right? So basically, we perform experiments in which we induce these animals with a reporter fluorescent protein, and we expose them to low doses of ionizing radiation. When I keep talking about low doses here, we talk about 50 milligrams of ionizing radiation, which is equivalent to 3 CT scans in the hospital. So really low doses of radiation. So we exposed the animals to ionizing radiation after being induced or being labeled, and then we basically analyzed the behavior of these clones. What we found is basically quite simple, that we increased the percentage of cells that were differentiating compared to those animals that were not irradiated, which means that basically ionizing radiation is pushing wild-type cells in our on this esophageal epithelium to differentiation. These numbers also correlate with a decrease in the basal cell density, because we promote more differentiation of the basal cells. When we count number of basal cells per area, we do find less basal cells in each area. So basically, what we were doing with radiation, with this low dose of radiation, was tilting this division outcome toward differentiation. Many basal cells in this tissue were differentiated. So basically, this is what we found using this model. We also tried to demonstrate that this increase in or this decrease in cell density in the basal was not mediated by apoptosis, because of the cells were dying, but differentiating. So we were checking, for example, caspase-3 in our tissues, and we found that low dose of radiation didn't trigger an increase in apoptotic cells in our tissues. But also we check of course, we're working with ionizing radiation, we need to study DNA damage responses. So we try to see whether we were inducing, or we were able to induce a significant amount of double strand breaks in the DNA using these low doses compared to typical high dose of 2 grays. And you can see here in this titration, is basically that 50 milligrams trigger a small increase in DSVs, but quite low compared to the high doses. We wanted also to analyze whether this increase in differentiation due to ionizing radiation was basically mediated by the immune system. In order to rule out that possibility, we basically designed, which I will talk about this later, but basically we designed an innovative 3D system model to study tissue responses in vitro. And we found that when we expose mouse and human epithelial, esophageal epithelium cultures, 3D cultures, to ionizing radiation, to this low dose of radiation, for example, in this case, using the X-Rail CIX-2 machine, using a 10 millimeters lead filter to attenuate the dose, because we were working with a very low doses to deliver 50 milligrams accurately. We were able to see an increase in differentiation in vitro of both mouse and human esophageal epithelial 3D cultures, which means, and rule out the possibility that the immune system is playing a role in this behavior. So basically, this differentiation, or this radiation, or this differentiation mediated by radiation, it was a cell autonomous effect. We also checked the response, the DNA damage response in these tissues, and we found, for example, as I mentioned before, that although we have detected a small amount of DSVs in this radiation, we were not able to detect an actual DDR activation pathway in this low dose radiation samples, which basically means that the cells were not able to activate the DDR pathway. However, they were basically sensing radiation and they were differentiating. So we were trying to see and analyze how this differentiation process was happening if we didn't have the increase in DSVs. One of the main, the other sources of, let's say, damage because of radiation is not only DSVs, but the main source can be oxidative stress. So basically, reactive oxygen species. So we know that there is one transcription factor, which is the master regulator of oxidative stress responses in the cells, which is called NRF2. And we know it mostly in normal conditions is bound to KIT1, and it's basically degraded into the proteasome in the cytoplasm. But when there is a change in the oxidative stress conditions in the cells, this NRF2 protein basically is released and goes into the nucleus and activates a bunch of genes, which are called basically the antioxidant response genes, which are able basically to deal with increases in oxidative stress. So we wanted to analyze whether our mice were basically increasing the levels of NRF2, or basically translocating NRF2 into the nucleus after being exposed to radiation. And this is what we checked. And what you can see here basically is that. So basically, low doses of radiation were basically increasing the levels of NRF2 translocation into the nuclei. So the cells were sensing radiation, and they were basically translocating NRF2 into the nucleus. So this response basically was making the cells to differentiate, or probably were making the cells to differentiate. We also analyzed the behavior we also analyzed how low doses of radiation are affecting not only wild-type cells, because all the studies we have done before was meant to be analyzing wild-type cells. But as I mentioned before at the beginning of the talk, you know that we are basically populated by mutant cells. So we wanted to analyze how these mutant cells that we know we have in our tissues are responding to ionizing radiation. So for that, we use a published model from Murray et al. In cell stem cell, which basically allows us to induce a reporter protein, but also it allows us to make a mutation, a hotspot mutation in the p53 gene. And this hotspot mutation, which is the R245W, which is equivalent to R48W in humans, is a very typical hotspot mutation found in squamous cell carcinomas. And we know that many tissues, especially the esophagus, have between 5 and 10% of these mutant cells in normal tissues. So we wanted to analyze how the effect of ionizing radiation can change the behavior of these cells in vivo. So we use this animal to induce the reporter protein, a GFP protein, but keeping in mind that this protein is also inducing the iteration of this hotspot mutation in heterozygosity, as we have in normal tissues. So basically, we did the same experiment. We induced the animals and we exposed the animals to ionizing radiation. And on the contrary, what happened in the normal cells, as I mentioned before, that basically wild-type cells tend to differentiate when we expose them to ionizing radiation. You can see here that after exposed p53 mutant cells in green to several doses of low dose of ionizing radiation, you can see how the p53 mutant population expand instead of being differentiated. So basically, we increase the pool of mutant cells in the animal, in the tissue, in the esophagus of the animal upon being irradiated. We were basically tracing and calculating different parameters showing how this mutation or these cells or the cells harboring this mutation basically behave completely different compared to wild-type cells. The idea was to understand how this mutation or this population is basically, why this population is behaving so different compared to wild-type cells when they are exposed to the same environmental factor, which is ionizing radiation. To do so, and keeping in mind that we believe that NRF2 or the antioxidant response might be playing a big role in these responses, we try to analyze how the ROS levels of in these two populations, in the wild-type population and in the p53 mutant population, were playing a role. To do so, we use a very well-known raw GFP2 transgenic reporter protein, which gives us specifically the amount of growth within each of mitochondria of the cells of these two populations. So we basically expose these cells to different doses, to this dose of radiation, low dose of radiation, and we measure these levels of oxidative stress in each mitochondria, as you can see here in this picture in green. So basically, you can here basically see in yellow the wild-type population of the mitochondria of the wild-type cells levels of oxidative stress upon being exposed to one single shot of 50 milligrams. You can see how the median of the oxidation goes higher, which basically is promoting the differentiation of wild-type cells. However, in the case of the p53 mutant cells, you can see here how the mutant population basically does not upregulate the oxidative stress and get, quite low compared to the wild population. So it seems that p53 population doesn't increase oxidative stress or ROS levels after being exposed to ionizing radiation. And we wonder whether this might be the cause why this population is basically resistant to radiation. We also analyzed by Aronistic data, and you can see how while wild-type cells downregulate and upregulate or change the pattern of expression of many genes upon radiation, which is a very typical sign of responding to ionizing radiation. You can see how this pattern in the p53 mutant population is basically completely black. So there are not many genes responding to ionizing radiation in this population. It seems that p53 mutant cells do not realize about being irradiated. We also analyze different pathways, and we check, for example, differentiation pathway. And you can see here how upon radiation, wild type cells upregulate all the genes implicated in differentiation. However, in the case of the p53, you can see those of genes are completely down, which correlates with the fact that this population do not differentiate, but proliferate and expand in the tissues. One interesting thing we found was basically that p53 population had already activated the mechanisms that control antioxidant response, even before being irradiated, which basically means that this population was primed to deal with ionizing radiation even before encountering the environmental factor. So basically, that's what this basically gives to this population a strong advantage in comparison with wild type cells. But we thought maybe what is the way we can externally manipulate this competition mechanism and control the expansion of mutant p53 cells from the outside. So we thought that maybe if we have a different response in terms of antioxidant pathways in these two populations, if we can hit the wild type cells with antioxidants and boost their antioxidant capacity, we might be able to basically balance this competition between these two populations and make the wild type cells a bit stronger, competing with the p53 cells. And that's what we did. Basically, we induce the animals, we expose them to radiation. As you know, when we expose them, the population of p53 cells overexpand. However, we treat the animals with antioxidants. And then what we saw basically was the opposite, that the antioxidant, the animals treated with antioxidant, basically the mutant population was not able to grow, was maintained at low level or single induction level, which means that wild type cells in this condition were stronger and basically were able to outcompete p53 mutant cells. So basically, we were able for the first time to show that there is a way that externally designing interventions that can control or shape cell competition mechanisms in order to deplete or at least to control the expansion or the behavior of different mutant populations in our tissues. And basically, this has a strong potential, because if we are able to manipulate the tissues from the outside, avoiding the expansion of specific mutant populations, which we know are heavily represented in tumors, we might be able to reduce the chances or the risk of tumor development in the future. As you can see here, for example, this is just to show you that after treating with antioxidant, we were able to block the translocation of NRF2 in our wild type cells. So basically, we were making our wild type cells stronger in terms of differentiation capabilities. I wanted to show you also something we are developing in the lab, which basically is quite useful for us and for many other groups, which allows us to study cell competition mechanisms, but also tissue responses in vitro over long-term experiments. Because this system, which we call epithelioids, allows us the generation of mouse and human primary epithelial 3D cultures to study, in my case, for example, radiotherapy responses in vitro over long periods of time. Let me just show you quickly. This work has been already sent. This preprint has been sent for publication, so you can find it in bioRxiv. So basically, the system works in a way that we take advantage of the original wound healing capacity or regeneration capacity of the tissues, of the epithelial tissues, and we cut the tissue in pieces. We set up we place the tissue in a scaffold, and we see how the tissue in the scaffold start healing, start generating a halo of cells, which is called an explant. And basically, this halo is growing from the beginning, from the middle of the tissue, trying to close the wound. So after 15 days, you can see here a confocal view. What we have is basically a whole well, fully confluent of cells coming from the initial piece of tissue. So one interesting thing is that this membrane, which is very simple, it means it has a strong effect on the maintenance of stem cells in these tissues. So basically, we can maintain these cultures for so long because we maintain the stem cell or the progenitor population of these original tissues. This is basically how a typical human esophageal epithelioid looks. You can see you have basal cells expressing alpha-6 integrin proteins in the bottom, and these cells are the ones proliferating and maintaining the culture. And we have differentiating cells that are migrating to the top and basically forming different layers. And this is quite similar to the actual tissue we have in vivo. This system allows us to study cell competition mechanism over longer period of time, because we can maintain this culture for, in the case of the mouse, up to 2 years in continuous culture. So we can study long-term processes in using this system. This is how the mouse esophageal epithelial looks. You can see here also proliferation and differentiation levels. So basically, with this system, we have been able to analyze or to characterize up to 12 months, although we have also some culture that we can maintain up to 2 years in the case of the mouse esophagus. And you can see how proliferation and cell density is nicely maintained over this period of time, over a year, which means basically the tissue is able to be maintained in a quasi-homeostatic conditions compared to other type of in vitro cultures that we know cells keep proliferating all the time and need to be passaged. When we analyze the transcriptional program of these two, of this system, of these 3D cultures, you can see, for example, the typical 3D view of the normal esophageal tissue and the epithelioid. You can see differentiation layers are quite similar. And when we study the transcriptional program, you can see that most of the genes follow a strong correlation between in vitro and in vivo, which means that this system is pretty reliable in terms of being used as a model to study tissue responses in vitro. We can generate epithelioid out of different type of mouse tissues. We can generate from tongue, from oral, vagina, trachea, uterus, many type of epithelial tissue. The only requirement we need for this generation is that the original tissue had or have regeneration capacity in order to set up the culture. This, of course, this epithelioid, they have a strong 3Rs impact, because we basically reduce hugely the amount of animals used in research, as we basically take advantage of this regeneration capacity. And for example, in the case of one single esophagus, we can generate the area equivalent to 182 esophagi, which basically impact tremendously into the 3Rs, basically the 3Rs. In this case, the two of them, so reduction, but also replacement, because we don't need to use also mouse tissues, because we can use human samples or human biopsies from donors as well. Again, using the X-Trail CIX1 benchtop irradiator, we are exposing our cancer epithelioids to different doses of radiotherapy every day, mimicking the same type of clinical treatment patients are receiving in the hospital. And you can see here, for example, how a tumor, which is basically a control, so it's not irradiated, you can see how over 2.5 months of time, the tumor overtake the whole culture and basically expand. However, when we use a typical radiotherapy treatment pattern, you can see how we can basically control the expansion of the tumor as is happening in real life. And we can basically, this is a proof of concept to show that our model can replicate the actual tumor behavior in vivo. This is basically typical confocal microscope use of this type of cultures. One of the projects, and this is just to finish my talk, we are quite interested in is understanding these novel radiotherapy treatments, which are really promising and basically are trying to use the flash effect, which is basically using ultra high doses of ionising radiation in radiotherapy treatments. So this flash effect, that you know, we are using normal conventional dose rate in the clinic to treat tumors in the patients, which are basically around 0.03 grays per second in typical radiotherapy treatments. And using these doses, we can control sometimes the tumor evolution, but we also produce quite huge toxicity into the normal tissue. Using this flash radiotherapy, ultra high dose, which basically can go up to 1,000 grays per second in the case of in the case of photons, basically allows us to control tumor expansion. We can basically kill the tumor, but it seems that we are not affecting, or at least not affecting so much, the normal tissue. So this effect is really, really interesting, and there are many groups in the UK and all over the world working trying to understand this flash effect and how this can be basically, once everyone understands how it works, basically translate into the clinic, because the benefit for the patient will be huge, because basically using such a high doses in a very short period of time, we will reduce the amount of time the patients are spending into the clinic. But also it might be quite beneficial, because we might kill the tumor easily and faster than with a conventional treatments. So this project basically is a three parties collaboration between Cambridge University, Manchester, and Oxford University. And we have a postdoc in my lab, which is basically working on this project, thanks to CIUK, RadNet, Cambridge funding. And basically, we are trying to set up these cancer epithelioid models and using this cancer epithelioid to understand how the tumors and the normal cells differentially respond to normal conventional radiotherapy and to flash radiotherapy. And basically, this is just to the acknowledgement to. I want to thank my lab, which is basically the people which are working on all these projects, and the people which are basically working hard to get all of them. Basically all the funding bodies and collaborators, especially CIUK RadNet, as Charlotte Coles and Nicola Leblon, the Gurdon Institute, of course, and Maria Colea and Inigo, which are one of the two main collaborators, which are basically making possible all these science to be done. I really want to thank X-Rail for all the help and support they have been provided to my lab. We are, as you have seen in my presentation, working with the CIX1 and the CIX2. Especially CIX1 is now giving us strong results, strong accuracy, and we can replicate all the experiment nicely, because the doses we are exposing these cells to are really accurate. So basically, I really want to thank the company for their strong support and all the help they are providing us in terms of ionization radiation capabilities. And thank you all of you for attending, and I'm very happy to take any questions if you have any. Thank you.
Thank you so much, Dr. Fernandez-Antoran. I appreciate that detailed presentation about your group's research. Very interesting, especially the interventions with the antioxidants. Just as a reminder to the attendees, if you could please submit your questions in the GoToWebinar control panel. I will go ahead and moderate those questions for Dr. Dr. Fernandez-Antoran. And let's go ahead and get started. There are a few questions coming in. Why did you choose the 50 milligram dose for the protocol?
Well, that has some different answers. So the first one is because I started this project being funded by an FP7 Euratom fellowship, and the idea of the funding body was to understand the effect of low doses of ionization radiation. So we decided to go for the lowest dose we can deliver in the lab, but in an accurate way. So we were able to deliver accurately up to 50 milligrays using a very complicated, because we were using a very old cesium source we have back in the 2013 at the MRC Cancer Unit before I met X-Rail company. And basically we had to use a 40 kilograms lead plates to deliver these 50 milligrams accurately. That was the reason why we chose this dose. And actually it's very interesting because there is no much research done on the effect of these really small doses in living tissues. That's the reason why we choose 50.
Okay. Next question, how do you expect your findings will depend on the photon energy used?
Sorry, I didn't pick the question.
The question is, how do you expect your findings will depend on the photon energy used?
So that's a very good point, because actually using flash, we are working with different type of LAT energies of the radiation, and depending on the type of LAT we use, we have different results. So it's highly depending on the LAT. Yes, I totally agree. But that's a very complicated thing, and it would be a different talk to give now to talk about this. But it's true. That's a very good point. It's very different. Actually, not only LAT, but also the type of beam we're using. We have seen different results using electron, using photons, and using protons as well. So it's very different responses of these tissues depending on the type of beams we are using to irradiate them. Yes.
Okay, next question. Are you looking at any other interventions besides antioxidants to control expansion?
Yes, we are looking now for a more targeted therapies, basically trying to target specific oncogenic mutations in the tissues in order to make them less fit to be, let's say, outcompeted by normal cells. So we are now working for different specific targeted therapies in order to deplete all the different mutations. Yes.
Okay. Next question, can you avoid the DDR with a higher dose than 2 gray, but a low dose rate less than 1 gray per minute?
No, we cannot. For example, we are trying now to avoid, because radiation is basically having in our hands two big effects in the tissues. So on one side, I'm talking about radiotherapy doses, so high doses of radiation. We're having activation of DDR pathway directly by DNA damage, but also we're having a huge production of ROS species. So we can block the production of ROS species with antioxidant directly, and basically we can detect the actual direct DNA damage we have because of the radiation, but we cannot do other way around so far. If someone has any ideas, I'm very happy to listen and to discuss.
Okay, next question. Can you explain more about the scaffolds and how they work?
Yes, sure. The scaffold is the critical part in this epithelial. Although it's very simple membrane, it's a pore membrane. The key here is the mechanics of the membrane. So basically, the number of pores per square millimeter and the size of those pores. So it seems that the cells attach to the membrane in a way that activate, different pathways that maintain the stemness of these cells. So when we change the pore size, we change the pore density, we do see completely different behavior in the cells, and we are not able to maintain these cultures for such a long time. Yes, it's a plastic membrane basically. It's not very fancy membrane or having any specific 3D structure. It's a plain membrane just full of pores.
Okay, thank you. Can you describe more about how your 6.1 and 6.2 systems complement each other?
Sure. So I must say I'm in love of the CI1 because it's a very small benchtop irradiator, and it's my favorite one. But we started having the CX2, and basically the number 2 is much more powerful and allows you to run more experiments, because it's also very well how do you say designed in terms of you can run heavy duty radiation, so you can do plenty of plates and run them for a long period of time. We also have a heater, so which allows us to maintain the temperature within the cabinet, so we can keep radiating the cells for longer periods if needed. And the CX1 allows us to keep doing daily dose radiations, basically. The normal 2 grays radiation, we are exposing the cells like mimicking the clinic treatments. So we can basically replicate that easily and really fast with the CIX1, because also the warming time and all the previous steps we have to do with the machine are much shorter. So it's quite fast and reliable. So this is how we play with these two type of equipment. Yes.
Okay, thank you. Can you tell us more about the flash radiotherapy and what factors you're considering?
Oh, flash radiotherapy is an amazing topic. My lab is now basically getting really involved. And the point about flash radiotherapy is very simple. It's basically increasing the dose rates of radiation up to levels in which everyone would think we are basically burning the tissues. But actually, the effect is actually the opposite. So we are maintaining the tumor killing effect, but we are sparing the normal tissue. The problem is nobody knows exactly how it works. There are different theories. People are talking about oxygen depletion, the effect on the immune system, because we keep in mind that we are delivering the same total dose, but the time in which we deliver the dose is very small. We're talking about microseconds. So we are exposing the tissues to a very short, sharp dose of radiation. So basically, now we're trying to understand how this flash is working, whether it's going through oxygen depletion, is going through the immune system component, and how we can this basically translate to the clinic in terms of helping patients to be treated with radiotherapy. And we're considering different factors because we are working with, in the same way we're working with conventional radiotherapy using proton, electron, and photons. We are also working with these type of beams in the flash field. So we're using proton flash, electron flash, and photon flash. And they allow us to deliver different dose rates because of the machines we're using. So in the case of the proton flash, we can deliver up to 40 grays per second maximum flash dose. And we do see the flash effect in the proton with 40 grays second. But in the case of the, for example, the electron, we can go up to 1,000 grays per second, which is insane, I must say. But basically, we try now to understand up to how much dose rate we can reach in order to have a safe flash effect without basically, affecting the normal tissue.
Okay, thank you. There are a number of comments just coming in complimenting you for the presentation and how much information you were able to cover in a short period of time. So thank you again for participating today. I think the audience has really enjoyed the presentation. Final question is around your epithelial cultures. Can you describe more about how you grow the epithelial organic cultures?
So it's a very simple system, although it might seem complicated. So basically, we take advantage of the actual regeneration capacity of the tissues. So a typical wound healing effect, when we have a cut in the skin, for example, the skin tries to heal. So when we basically cut a piece of tissue, either a patient biopsy or a tissue from the animal, and we cut it in pieces, we are triggering this regeneration or this wound healing effect. So we are taking advantage of this wound healing effect, and we are placing this tissue in a scaffold in which this healing can be basically forming a new tissue. That's all. That's how we do it. And basically, given the nature of this scaffold, this new tissue which is generated is self-maintained. So we can maintain the culture for such a long time because of that, because the tissue itself is self-maintained as normal tissues in the body.
Okay, all right, thank you again. On behalf of Extral, we'd like to thank the attendees, and of course, thank you so much, Dr. Fernandez-Antoran, for your presentation today. As a reminder, you will receive a recording of this event via email after the recording has been posted to the website. With that, I'll go ahead and conclude today's session, and thank you so much for everyone's attention today. We really appreciate it. Have a great afternoon. Bye-bye.
Thank you. Bye.